everyone, a very good morning to everyone. So I am Bushra, your botany faculty, and today I am going to deal with a topic which is from the chapter photosynthesis in higher plant, which is from the 11th class, and the topic which I am going to deal today is C3 versus C4 plant. It is one of the most important topic from exam point of view as well as economical as well. वो आप कैसे वो हम क्लास में देखेंगे, right? So very good morning. So today we are going to uncover the process that fuel the life on the earth, which is C3 pathway and C4 pathway, due to which two types of plants are there. Those are C3 and C4. So students, as you know that the process of photosynthesis is there, which involves the carbon dioxide from the atmosphere, which combines with water in the presence of sunlight in the chloroplast of the green leaves, and it will lead to the formation of glucose with the liberation of oxygen. अगर हम normally बात करें तो photosynthesis दो process में divided है. First one is light reaction, next one is dark reaction. Simple, right? Let's see. It is divided into two processes. Those are light reaction and dark reaction. जैसा कि नाम में खुद ही है कि light reaction in the presence of light, but dark reaction, it does not mean that it occur only in dark, right? It is actually the misnomer. Why? Because it utilizes the products formed in the light reaction, but it is not directly dependent on the light. Okay, so light reaction involves certain steps, for example, absorption of light, splitting of water, and liberation of oxygen. It consists of cyclic and non-cyclic photophosphorylation, which is followed by chemiosmotic hypothesis, right? And now the products which are formed, or we can say the assimilatory power. The assimilatory power, which are ATP and NADPH, are utilized in the dark reaction for the formation of food. You know that light reaction takes place in the thylakoid membrane and dark reaction takes place in the stroma. Okay, so here we are going to deal with the dark reaction specifically. How? Let's see. So in dark reaction, as we are going to utilize the assimilatory power formed in the light reaction, those are ATP and NADPH. Now plant have to assimilate the carbon dioxide from the atmosphere and utilize it for the formation of carbohydrates. On the basis of different approach for assimilating the carbon dioxide from the atmosphere, the plants are divided into three types. Those are C3 plants, C4 plants and CAM plants. अब ये ऐसा क्यों है? C3 plants, C4 plants and CAM plants, they are differentiated on the basis of different types of metabolic pathway for assimilating the carbon dioxide. But one thing is common in them, that is Calvin cycle. Calvin cycle is also known as C3 cycle, right? तो ऐसा नहीं है कि C3 plants में सिर्फ Calvin cycle होती है और C4 plants में सिर्फ hatch and slack pathway होता है. C3 cycle takes place in all the three types of plants, C3, C4 and CAM plants, or we can say Calvin cycle takes place in all the photosynthetic plants. Okay. In C3 plants, only C3 cycle takes place, and if we talk about C4 plants, along with C3 cycle, hatch and slack pathway is also there. Why? because it has adapted to certain conditions so that it can assimilate the carbon dioxide from the atmosphere efficiently. इसमें कुछ कमियां रह गई होंगी. To adapt the adverse condition, they have adapted certain anatomical features which help in assimilating the carbon dioxide more efficiently. Same way in the CAM plant also, उसमें भी कुछ anatomical modifications हुए और उसमें carbon dioxide assimilation में modify हुआ और pathway बना. That's why they are differentiated into 3 types of plants. Now let's see that how they actually look like. C3 plants have a very simple anatomical features. That means they have a mesophyll cells. Mesophyll cells जिसमें abundant amount of chloroplast present होता है और यहाँ पे Calvin cycle हमारी चलेगी. Now let's move to the next plant, that is C4 plants. It has very beautiful anatomical feature which is known as Kranz anatomy. जैसा कि हमने C3 plant में देखा that it has the mesophyll cell. These are the mesophyll cells and this is the vascular bundle. But in the Kranz anatomy, which is specifically, or we can say the characteristic feature for the C4 plants, that vascular bundles are surrounded by bundle sheath cells. So these are the vascular bundles and they are, yes, good morning everyone. So they are surrounded by bundle sheath cells. These bundle sheath cells are thick-walled. They are impervious to gaseous exchange. उसमें कोई भी gas in and out बहुत easily नहीं हो सकती. That means vascular bundles are tightly packed via the layer of bundle sheath cells, and later on these are the mesophyll cells which surround it. So it is ring-like pattern, which is also known as wreath arrangement. So it is the characteristic feature for the C4 plants. Now if we move to the third type of plant, that is CAM, that is crassulacean acid metabolism plant. In them large number of mesophyll cells and vacuoles are present which are of large size. Why? Because they have to accumulate the acid in their vacuole for further assimilation of carbon dioxide in its cells. Right? So these are the anatomical difference between C3, C4 and CAM plants. Now let's move to the C3 plants. In C3 plants, as I've told you, the common feature among all of three of them is Calvin cycle. So let's first see that how Calvin cycle runs. A very brief intro for the Calvin cycle. Why it is named so? It is after the name of the scientist and it is also known as C3 pathway. Why C3 pathway? Because the first stable compound which is formed during the Calvin cycle is 3-carbon compound, which is phosphoglyceric acid. Right? Now let's see. The first carbon dioxide acceptor, or we can say the primary CO2 acceptor in the C3 plants is, or we can say in Calvin cycle is RUBP, which is a 5-carbon compound. Right? अब ये primary acceptor है carbon dioxide का, तो इसके पास already 5 carbon थे. अब इसने accept किया 1 molecule of CO2 from the atmosphere. In the presence of enzyme which is rubisco. Rubisco is expanded as ribulose, bis, phosphate, carboxylase and oxygenase. That means it has 2 different activities, carboxylase as well as oxygenase. Carboxylase, fixation of carbon dioxide, oxygenase, fixation of oxygen. But here, as it have to work as a carboxylase because यहाँ पे हम कार्बन डाइऑक्साइड को फिक्स कर रहे हैं. तो अगर हम देखें इसके पास 5 कार्बन है, यहाँ से एक कार्बन बन के 6 कार्बन का एक मॉलिक्यूल बनना चाहिए, बट द फर्स्ट स्टेबल कंपाउंड इज एक्चुअली 3 कार्बन phosphoglyceric acid. So once the 6 carbon molecule is formed, it get dissociate into 2 molecules of 3PGA, phosphoglyceric acid, which is 3 carbon molecule, right? Now as you can see that phosphate is attached to this group. अब ये phosphate कहाँ से आए? Because जो हमारा primary CO2 acceptor था, which is ribulose bisphosphate, यानी कि इसके पास 2 phosphate group थे, so it's donated to the phosphoglyceric acid and it is forming phosphoglyceric acid. 2 molecules बन गए, right? And this process is known as carboxylation. Right? Fixation of carbon dioxide into the cell. Now from this, next step will take place. ये acidic group है, that means COOH लगा हुआ है. It is highly oxidized form. अब हमको इसको reduce करना है, that means we have to remove the oxygen and add the hydrogen in them, right? अब इसका reduction होगा, this process is known as reduction. For this process, for reduction of 3-phosphoglyceric acid, there will be involvement of 2 molecules of ATP along with 2 molecules of NADPH. जैसा कि हमने देखा था कि ATP और NADPH हमारे light reaction में बने थे, so it is utilizing here. 2 molecules of ATP and NADPH are utilizing in the reduction step of Calvin cycle, right? So here a 6 carbon molecule will be formed from where 1 carbon will get utilized for the formation of glucose और कार्बोहाइड्रेट. अब ग्लूकोज में कितने कार्बन होते हैं? 6, राइट? तो एक साइकिल से अभी सिर्फ एक कार्बन ही आ पाए बिकॉज इट हैज फिक्स्ड ओनली सिंगल कार्बन डाइऑक्साइड फ्रॉम द एटमॉस्फेयर. दैट मीन्स टू फिक्स द a molecule of glucose, this cycle have to run 6 times, right? Now this यहाँ से हमारा एक कार्बन चला गया for the formation of glucose. The remaining 5 carbon will lead to the regeneration of the primary acceptor, that is RUBP, which is of 5 carbon. This process is known as regeneration. For the regeneration, again a molecule of ATP is utilized. Right? So here you can see that 3 molecules of ATP along with 2 molecules of NADPH are utilized for the fixation of single carbon molecule, and if we calculate for the single glucose molecule, that means it consists of 6 carbon. That means 3 into 6, 18 ATP and 12 NADPH are required for the fixation of single glucose molecule in the C3 pathway. Okay, and this complete pathway is taking place in the cell which is mesophyll cell. जैसा कि बताया था कि C3 plants में जो कार्बन का assimilation है वहाँ पर सिर्फ सिंगल सेल इन्वॉल्व है, that is mesophyll cell. राइट? सो यहाँ से हम लोग अपने डिफरेंस लेना स्टार्ट करेंगे. यहाँ पर जो assimilation है, primary assimilation of carbon dioxide is taking place in the mesophyll cells. The first stable compound is 3 carbon, that is phosphoglyceric acid. The primary carbon dioxide acceptor is RUBP, which is 5 carbon compound, and these are the features of C3 pathway which is taking place in the C3 plants. Now let's move ahead and see the differences between them. Okay, yeah. So these are the difference between the C3 and the C4 plants. The first product is 3 carbon. Later on, when we study about the C4 plants, we will see that the first product is 4 carbon containing compound. Next, the photosynthesis site, as I've told you that in C3 plants it is mesophyll, and in C4 another cell will get involved in it. Next one is Kranz anatomy, as I've told you about the anatomical feature of C3 and C4, where the characteristic feature of C4 plant is presence of Kranz anatomy, which is also known as wreath arrangement, which is found in the C4 plants. Now next is photorespiration. अब ये एक वेस्टफुल प्रोसेस है, हम आगे पढ़ेंगे कि किस तरीके से होता है. जैसे कि अभी हमने देखा दैट रुबिस्को का कार्बोक्सिलेज एक्टिविटी थी, तब जाके उसने C3 पाथवे को रन किया. व्हाट इफ ऑक्सीजनेज एक्टिविटी हो, राइट? सो वो हम अभी देखेंगे फर्दर क्लास में. नेक्स्ट इज ऑप्टिमम क्लाइमेट. अगर हम C3 प्लांट्स की बात करें, सो दे जनरली अकर इन द कूल, मॉइस्ट एंड नॉर्मल लाइट कंडीशन. इनको ज़्यादा गर्मी सहन नहीं होती, ये ज़्यादा गर्म एनवायरनमेंट में नहीं अच्छे से ग्रो कर पाते. इनका रेट ऑफ फोटोसिंथेसिस कम हो जाता है, एफिशिएंसी कम होगी, एंड ऑब्वियसली फिर यील्ड और प्रोडक्टिविटी पर अफेक्ट पड़ेगा. वहीं पर ही अगर हम C4 प्लांट्स की बात करें तो वहाँ पर ऑप्टिमम क्लाइमेटिक कंडीशन है हॉट एंड ड्राई. जितना ज़्यादा टेम्परेचर होगा तो भी ये अपना सर्वाइव कर लेगा. दैट मीन्स एट हायर टेम्परेचर एंड इन द लेस वाटर कंडीशन और स्कार वाटर कंडीशन it can survive well. Next is tomato. If we talk about the C3 plants, C3 प्लांट्स में स्टोमेटा जो हैं वो ज़्यादा देर तक ओपन रहते हैं. वाई? क्योंकि उनको CO2 लेना है. अब CO2 लेना है तो उसके वजह से जो वाटर वेपर्स हैं विच इज़ ऑलरेडी प्रेजेंट इन द प्लांट गेट लॉस बाय द प्रोसेस ऑफ ट्रांसपिरेशन, राइट? बट अगर हम C4 प्लांट्स की बात करें तो वहाँ पर जो स्टोमेटा ओपनिंग का टाइम है वो काफ़ी कम है, बिकॉज जिसके वजह से वो वाटर कंजर्व कर पाते हैं और तभी ये वाटर स्कार्सिटी कंडीशन में भी सर्वाइव कर पाते हैं. अब ये ऐसा क्यों है? क्या इसको कार्बन डाइऑक्साइड की ज़रूरत नहीं है? बिल्कुल है, बट ये कार्बन डाइऑक्साइड को किस तरीके से एसिमिलेट करता है वो हम आगे देखेंगे. नेक्स्ट वन है CO2 fixation enzyme. Here the rubisco is only involved, but in the C4 pathway pep carboxylase along with rubisco are involved. Next one is efficiency. C3 plants are less efficient at higher temperature, and if we talk about the C4 plants, they are more efficient. Next is example. C3 प्लांट्स की अगर हम बात करें तो मोस्ट ऑफ द क्रॉप्स आर ऑफ C3 प्लांट्स, राइट? फॉर एग्जाम्पल द लेग्यूमिनस प्लांट्स वो सारे C3 होते हैं. हमारा वीट, राइस, सोयाबीन एंड कॉटन, ऑल दीज आर C3 प्लांट. और हमने अभी देखा कि उनकी एफिशिएंसी कम है. वाई? बिकॉज हायर टेम्परेचर वो कम एफिशिएंट होते हैं. कैसे होते हैं? लेट्स सी फर्दर. नेक्स्ट C4 प्लांट्स की अगर हम बात करें तो मेज एंड शुगर केन, sorghum, millets, these are the example of C4 plants. And if we talk about the chloroplast type, only one type, that is mesophyll cell, is involved. And if we talk about the C4 plants, here 2 cells are involved. Those are mesophyll cell and bundle sheath cell. Now let's see the next thing, which is C4 pathway, that is Hatch and Slack pathway, which is taking place in the C4 plants. Why they are named so, C4 plants? जैसा कि हमने C3 में देखा था that first stable compound which is formed in the C3 plants is 3 carbon containing, that is phosphoglyceric acid. वैसा ही यहाँ पर जो first stable compound है that contains 4 carbon, that is oxaloacetic acid, OAA, which is of 4 carbon. Right? Now let's start the cycle. Before that I would like to tell you that it involves 2 cells. अब इसमें 2 cells कौन से हैं? One is mesophyll cell and another one is bundle sheath cell. हमने देखा था Kranz anatomy में bundle sheath cells present है which are surrounding the vascular bundles. Now let's make the cells. So ये हमारा बन गया mesophyll cell. Mesophyll cell. Right. अब यहाँ पे भी एक primary CO2 acceptor होगा, which is PEP. What is PEP? Phosphoenol pyruvate, which is of 3 carbon. अगर हम C3 की बात करें तो वहाँ पे जो हमारा primary carbon dioxide acceptor था, which is of 5 carbon, that is RUBP. यहाँ पर हमारा PEP है. Right? Now it will fix 1 molecule of CO2 from the atmosphere in the presence of enzyme PEP carboxylase. I am writing here PEP carboxylase. So the enzyme which is involved in the fixation of carbon dioxide from the atmosphere in the mesophyll cell is PEP carboxylase, phosphoenol pyruvate carboxylase. C3 plants में हमारा Rubisco था, right? While fixation, यहाँ पे 3 carbon था, इसका 1 carbon आया, so here it will forming 4 carbon compound which is oxaloacetic acid, which is the first stable product formed in the C4 pathway. Now, this 4 carbon compound cannot move to the bundle sheath cell as such. यहाँ पे हम bundle sheath cell बना देते हैं, and these are connected via plasmodesmatal connections. So, ये 4 carbon compound will get converted into another 4 carbon compound. It could be of malic acid, या तो malic acid या malate में बन जाएगा, या फिर aspartic acid या aspartate में ये convert हो जाएगा. Right? Now, this acid will get transported to the bundle sheath cell. This is 4 carbon compound which get transported in the bundle sheath cell. ये हमारा bundle sheath cell है. अब यहाँ पे 4 carbon compound है. What will happen next? Now the process of decarboxylation will take place. What is decarboxylation? Removal of carbon dioxide. यहाँ पर 1 कार्बन कम हो गया, that means यहाँ पर हमारे पास 3 कार्बन कंपाउंड बचा which is pyruvic acid. तो decarboxylation हो गया, जो first compound बना that is pyruvic acid, and this CO2 get into the process known as Calvin cycle. यहाँ पर हमारा CO2 assimilation हो रहा है where? In the bundle sheath cell. अगर Calvin cycle is taking place in bundle sheath cell, that means in the bundle sheath cell there will be presence of Rubisco enzyme in abundance, but it is absent in the mesophyll cell of C4 plant. This is also one of the difference. In C3 plant, Rubisco enzyme कहाँ था? Mesophyll cell में. But C4 plant में Rubisco enzyme है bundle sheath cell में, because Calvin cycle is taking place in the bundle sheath cell. अब ये 3 carbon compound which is pyruvic acid will again go into the mesophyll cell and regenerate the phosphoenol pyruvate, which is the primary CO2 acceptor of C4 pathway. So this is the complete cycle. अगर हम steps की बात करें, वहाँ पे 3 step थे, यहाँ पे भी 3 step हैं. First हमारा हो गया carboxylation, fixation of carbon dioxide. Let's jot down here carboxylation. Next step हमारा हुआ bundle sheath cell में, which is decarboxylation. decarboxylation. And third step is regeneration from pyruvic acid to the phosphoenol pyruvate in the mesophyll cell. Like this. तो अगर हम बात करें टोटल अमाउंट ऑफ एनर्जी और एसिमिलेटरी पार्ट इसमें कितना यूज हुआ, तो यहाँ पे केल्विन साइकिल तो हो ही रही है. अलॉन्ग विद दैट one more pathway is taking place, that means fixation of CO2 in the 4 carbon compound and then it is transporting to the bundle sheath cell. So yeah, so here how many ATP are required? So 3 ATP are already utilized in the Calvin pathway which is taking place in the bundle sheath cell. I am writing here 2 NADPH and 3 ATP. Along with that, extra 2 ATP are required for regenerating the PEP in the mesophyll cell. तो अगर हम total बात करें, तो here in the Hatch and Slack pathway, 5 ATP along with 5 ATP along with 2 NADPH are required for fixation of single CO2 molecule. Again, if we talk about the formation of glucose molecule which consists of 6 carbon, then it will be requiring 30 molecules of ATP and 12 NADPH, which is far way more than the C3 pathway. But if we look into the efficiency of the plant, C4 is much better. Why? Let's see. जो हमने C3 प्लांट पढ़ा था उसमें C3 पाथवे था, सिर्फ C3 पाथवे था, तो उसके कार्बन डाइऑक्साइड फिक्सेशन के लिए the plant have to open its stomata for longer period of time. Why so? To accept the carbon dioxide, due to which the water vapor get evaporated from the plant cell, which will lead to the water scar condition in the plant cell. तो यानी कि वो ज़्यादा hot and humid hot condition में नहीं survive कर पाएगा, उसमें water की condition कम, water का amount कम हो जाएगा. वहीं अगर हम बात करें C4 प्लांट्स में जिसमें हैच एंड स्लैक पाथवे हो रहा है, यहाँ पे उन्होंने कार्बन डाइऑक्साइड को फिक्स किया इन द मेसोफिल सेल एंड लेटर ऑन they conserve their carbon dioxide into the 4 carbon compound which is malic acid or aspartic acid, यानी कि उनका रिजर्वोयर बन गया for the carbon dioxide. कार्बन डाइऑक्साइड कहाँ जाके टूटा? इन द बंडल शीथ सेल. तो यहाँ पे CO2 कॉन्सेंट्रेटिंग मैकेनिज्म है एट द एंजाइम साइट, जो कि रुबिस्को की एंजाइम साइट है, जिसको कार्बन डाइऑक्साइड का कॉन्सेंट्रेशन हाई चाहिए टू मेंटेन इट्स कार्बोक्सिलेज एक्टिविटी. Right, to maintain its carboxylase activity, the concentration of CO2 must be higher in the bundle sheath cell of C4 plant, due to which उसको ज़्यादा देर के लिए अपना स्टोमेटा ओपन करने की ज़रूरत नहीं है. That means the water evaporation or transpiration rate will be decreased, right? That's why it is showing good or better efficiency as compared to the C3 plants. Okay, so these are the differentiating features between C3 and C4 which will be jot down later, or let's jot down right now so that we can remember in future as well. Now let's make the difference table. ठीक है, तो अगर हम बात करें प्राइमरी CO2 एक्सेप्टर की, सो इन C3 it is 5 carbon RUBP, ribulose bisphosphate. If we talk about C4 plant, the primary CO2 acceptor is 3 carbon PEP, phosphoenol pyruvate. The first stable compound which is formed in the C3 plants is 3-carbon compound, that is phosphoglyceric acid, PGA. Please don't get confused with phosphoglyceraldehyde because this is acidic compound. Now in the C4 plant, the first stable compound is 4-carbon, that is oxaloacetic acid. If we talk about the cells which are involved in the C3 and the C4 pathway, so here only one cell is involved, that is mesophyll cell. And if we talk about the C4 plant, यहाँ पे उन्होंने अपने काम को divide कर दिया. There is division of labor, जिसके जो एक और कारण है कि वो क्यों ज़्यादा efficient है. So here 2 cells are involved along with the mesophyll cell. Bundle sheath cells are also involved. Now, if we see the activity of Rubisco enzyme, so in them, Rubisco enzyme get activated or it is present in the mesophyll cell of C3 plants. But if we talk about the C4 plants, in them, Rubisco, if we talk about the C4 plants, in them, the Rubisco enzyme is found in the bundle sheath cell. If we talk about the mesophyll cell, it contains PEP carboxylase, phosphoenol pyruvate carboxylase. Please remember, this is the very important differentiating feature in them. But in mesophyll cell, no Rubisco is present. And if we talk about bundle sheath cell of C4 plant, no PEP carboxylase is present. Right? Now, if we see the number of ATP which are required to fix the single carbon molecule, so here 3 ATP are required and here 5 ATP are required. If we talk about the number of NADPH, in both 2 molecules of NADPH are required. So that's how we can calculate the number of ATP and NADPH in both the plants for the formation of single molecule of glucose. Right? So these are the basic common differentiating features between C3 and C4 plants. Now let's move ahead. So this is crassulacean acid metabolism. ये NCERT में नहीं दिया है. I'm going to just give you the brief about the CAM pathway, that how it takes place. ये pathway उन plants में होता है जो कि arid region, or we can say this pathway takes place in the plant which are found in the arid region, or we can say the water scar condition or xerophytic condition में present होते हैं, जहाँ पे water है ही नहीं. For example, cactus or the plants belonging to class bromeliads. Right? तो यहाँ पर इतनी ज़्यादा heat होती है, the temperature may go beyond 45 degrees Celsius. So what they do, they temporal distribute their carbon dioxide assimilation. Temporal assimilation of carbon dioxide fixation in them takes place. How? यहाँ पे day और night में division of labor होता है. कैसे? Again, here mesophyll cells are involved. In CAM plants, what will happen? Carbon dioxide get fixed. The primary CO2 acceptor is PEP. Here, carbon dioxide get fixed in the presence of PEP carboxylase and get converted into oxaloacetic acid. Now, this oxaloacetic acid get converted into another 4-carbon compound, that is malic acid, malate or aspartate. And these get accumulated in the vacuole of the plant. I've told you initially कि कैम में क्या एनाटॉमिकल डिफरेंस है, इसमें लार्ज साइज ऑफ मीसोफिल सेल्स और लार्ज साइज ऑफ वैक्यूल प्रेजेंट होते हैं. तो यहाँ पे जो भी एसिडिक कंपाउंड बना वो जाके सेव हो गया हमारे वैक्यूल में, राइट. एंड दिस fixation of carbon dioxide is taking place during the night time. Why? दिन में तो बहुत ज़्यादा गर्मी होती है, the temperature go beyond the 45 degree Celsius. अगर प्लांट ने अपने स्टोमेटा खोले तो सारा वाटर वेपर, सारा वाटर इवेपोरेट हो जाएगा from the plant, right? And plant cell will die. So for that, नाइट में ये अपने स्टोमेटा ओपन करेगा, देन इट विल एक्सेप्ट द CO2 फ्रॉम द एटमॉस्फेयर एंड फिक्स इट इनटू द 4 कार्बन कंपाउंड malate or aspartate and get it saved in the vacuole. Now in the daytime what will happen? This aspartate, which is the 4 carbon compound, get dissociated into 2 parts. 3 carbon compound along with carbon dioxide. So this carbon dioxide will go into the Calvin pathway. So यहाँ पे हमारा carbon dioxide assimilate हो गया and this 3 carbon compound will regenerate the PEP. So this process or the Calvin cycle is actually taking place in the daytime. So this is the temporal separation of carbon dioxide fixation in the CAM plants. Now let's move ahead. Photorespiration. What is photorespiration? Light के presence में respiration हो रहा है. Simple as that. As I've told you, in the C3 pathway, the prime enzyme which is involved is rubisco. Rubisco का full form क्या था? Ribulose, bisphosphate, carboxylase and oxygenase. Carboxylase and oxygenase. जब C3 pathway run हो रहा था, at that moment carboxylase activity is shown by rubisco enzyme. But when there is presence of oxygen as well, and it will act as an oxygenase, that means it has higher affinity towards the oxygen, then photorespiration will take place. और अगर हम C3 and C4 की बात करें तो C3 plants में photorespiration होता है but C4 plants में नहीं होता. According to NCERT, that photorespiration does not take place in the C4 plants but it will take place in the C3 plants. Now let's see how. The rubisco enzyme वो अपना oxygenase activity show कर रहा है. That means it will combine with the oxygen. 5 कार्बन कंपाउंड है ये. यहाँ से उसको कार्बन तो मिला नहीं, ऑक्सीजन के पास कोई कार्बन नहीं है तो कोई कार्बन एसिमिलेट नहीं हो पाया and it will lead to the formation of 2 compounds. One is 3 carbon compound along with that 2 carbon compound. हमारा 3 कार्बन कंपाउंड सेम फॉस्फोग्लिसरिक एसिड ही है जो कि कैल्विन साइकिल में रन होगा एंड दिस 2 कार्बन कंपाउंड विच इज फॉस्फोग्लिसरेट. Right, this compound is actually toxic to the plant and it is of 2 carbon. This complete process is actually completely wasteful process क्योंकि यहाँ पे ना ही तो कार्बन का assimilation हो रहा है, ना ही कोई ATP और NADPH यूटिलाइज फॉर्म हो रहा है, बट उसके अलावा जो ATP और NADPH आपका बना था उसको भी यूटिलाइज कर ले रहा है. राइट, सो दिस फॉस्फोग्लिसरेट अब ये बन तो गया है तो इसको यूटिलाइज भी करना है. सो फॉर दिस प्रोसेस 3 डिफरेंट ऑर्गेनेल्स आर इन्वॉल्व. द फर्स्ट वन इज क्लोरोप्लास्ट. ये ग्रीन कलर का हमारा क्लोरोप्लास्ट है. नेक्स्ट वन इज peroxisome, जहाँ पे peroxides बनते हैं. And the third organelle which is involved is mitochondria. Please remember, these are asked a question in the NEET exam, right? So here 3 organelles are involved: chloroplast, peroxisome and mitochondria. तो phosphoglycolate और glycerate जो बना था हमारा chloroplast में, it will get converted into glycolate in the chloroplast itself. Then it will move to the peroxisome and it will lead to the formation of glycine, which is the amino acid. और यहीं पर ही हमारे peroxide भी बनते हैं, that is H2O2, right? While moving glycine from the peroxisome to the mitochondria, and then further this glycine getting converted into serine, which is the another amino acid. Here a molecule of NADPH is utilized. अब ये serine जो है, it will move from the mitochondria to the peroxisome, and here it will get converted into glycerate. Again here one molecule of NADPH will get utilized. Now this glycerate will again go into the chloroplast and will lead to the formation of PGA, and here a molecule of ATP is utilized. तो इस process में ना ही कोई ATP बन रहा है, ना ही कोई NADPH बन रहा है, ना ही CO2 फिक्स हो रहा है. फिक्स होने के बजाय from the mitochondria a molecule of CO2 get released. फिक्स भी नहीं हो रहा है, रिलीज हो रहा है, so it is a complete wasteful process in the plant. और ये किस में हो रहा है? It is taking place in the C3 plants. Why? Because उसमें rubisco enzyme अपना oxygenase activity show कर देता है when there will be deficiency of carbon dioxide, और carbon dioxide की deficiency होगी C3 plants में. ये ऐसा क्यों होगा? Let's see further in the class. ठीक है? So this is the complete wasteful process and it is taking place only in C3 plants. That's why इनकी efficiency, productivity and yield सब कम है. Now let's move ahead in the class where we will study about the factors affecting photosynthesis. अब photosynthesis का process हो रहा था where we are forming the carbohydrate molecule, that is glucose, which is required to sustain the life on this planet. Right? So there will be certain factors which are affecting the process of photosynthesis. Those factors could be external or internal. External में आ गए हमारे sunlight, temperature, etc. All these are external factors which are not present in the plant and affecting the rate of photosynthesis. Right? Now this is the graph for the light intensity. Now let's consider the light intensity along with the carbon dioxide concentration. अगर carbon dioxide concentration high है, let's consider CO2 concentration high and light intensity low. So in this condition, both C3 and C4 plants do not show any higher rate of photosynthesis. No higher rate of photosynthesis. On the contrary, जब हम CO2 की concentration भी ज़्यादा है along with high light intensity, then both C3 and C4 plants will respond and show high rate of photosynthesis. Acha, for the factors there is one law. which is known as law of limiting factors, which is given by Blackman. अब उन्होंने क्या बताया that there are multiple factors which are affecting the rate of photosynthesis, but there is some factor which is present in the least amount. For example, यहाँ पे हमने light लिया था, right? Light की जो intensity थी वो कम थी, and carbon dioxide concentration is higher. That means here in this condition, the factor which is affecting the rate of photosynthesis is intensity of light, not carbon dioxide. So जो factor कम amount में present होगा, that will decide the rate of photosynthesis. This is what the Blackman's law of limiting factor states, right? So likewise, light intensity, अब यहाँ पे हम ग्राफ में देखते हैं that the way we are moving or increasing the light intensity, the rate of photosynthesis is also increasing, but up to certain limit. After this limit, the rate of photosynthesis get constant with the light intensity. लाइट इंटेंसिटी और लाइट का सैचुरेशन प्लांट्स में कब आ जाता है? ओनली 10% ऑफ द सनलाइट, इतने ही परसेंट पे इसका लाइट सैचुरेशन आ गया, बिकॉज वी हैव अबंडेंस ऑफ लाइट. दैट मीन्स जो लाइट है इट विल rarely become the limiting factor because it is already present in abundant amount on this planet. So अगर हम लाइट की बात करें, so it rarely become the limiting factor. राइट यहाँ पे हमने देखा सैचुरेशन आ गया, एंड फर्दर अगर हम लाइट देंगे तो वहाँ पे क्लोरोफिल डिस्ट्रक्शन हो जाएगा. राइट, सो दिस इज ऑल अबाउट द लाइट. नाउ इफ वी सी about the carbon dioxide concentration, that how the carbon dioxide concentration will affect the rate of photosynthesis. कार्बन डाइऑक्साइड कॉन्सेंट्रेशन एक हमारा होता है एटमॉस्फेयर में, एक होता है हमारा इंटरनल कार्बन डाइऑक्साइड कॉन्सेंट्रेशन. राइट, अगर हम एक्सटर्नल और एटमॉस्फेयर कार्बन डाइऑक्साइड कॉन्सेंट्रेशन की बात करें, सो इन आवर एटमॉस्फेयर, अगर हम एटमॉस्फेरिक CO2 की बात करें, so we have only 0.03% to 0.04% of CO2 concentration in the atmosphere, right, which is around 380 microliter per liter. Right, ये equivalent है इतने के. And if we talk about C3 and the C4 plants, again let's see the difference between them. So if we see the C3 plant and if we see the C4 plant, so here the saturation of carbon dioxide in the C3 plants is beyond, that means greater than 450 microliter per liter. But if we talk about the C4 plants, the requirement for its carbon dioxide is 360 microliter per liter. तो अगर हम कंपेयर करें C3 और C4 प्लांट्स में, तो अभी भी कौन ज़्यादा डेफिशिएंट है कार्बन डाइऑक्साइड के? C3. Again, this is one of the reasons that why the productivity and efficiency of C3 plant is less than the C4 plant. क्योंकि इतना अमाउंट तो प्रेजेंट है ही नहीं एटमॉस्फेयर में, लेकिन still it is required beyond 450 microliter per liter of carbon dioxide concentration for its optimum utilization. That's why the productivity of C3 is less than C4 plants. राइट, अब यहीं पर ही एक और बात आ जाती है. 450 माइक्रोलीटर का इसका है कैपेसिटी की, इतना ले सकता है कार्बन डाइऑक्साइड, बट हमारे एटमॉस्फेयर में 380 माइक्रोलीटर पर लीटर ही है. तो अगर हम C3 प्लांट्स की बात करें, फॉर एग्जांपल टोमेटो एंड बेल पेपर, जैसा कि मैंने बताया था कि C3 प्लांट्स में बहुत सारे हमारे क्रॉप प्लांट्स भी आते हैं। C3 प्लांट्स में हमारे बहुत सारे क्रॉप प्लांट आते हैं, इसमें से हमारे टोमेटो और बेल पेपर हो गए। तो अभी तक इनका जो यूटिलाइजेशन है या कैपेसिटी है फुलफिल नहीं हो पाई है। एंड इफ वी प्रोवाइड द एक्स्ट्रा CO2 concentration, that means in the greenhouse, if we provide extra CO2 to increase its photosynthesis, then it will yield high productivity. Right? So increased CO2 concentration atmosphere में रखा गया इनको. That means in the greenhouse जहाँ पे ज़्यादा CO2 concentration था, जहाँ पे ज़्यादा CO2 concentration था, जिसके वजह से higher productivity हुई. This is known as CO2 fertilization effect. बिकॉज अभी तक इनका रिक्वायरमेंट फुलफिल नहीं हुआ था, हमने एक्स्ट्रा CO2 प्रोवाइड कराया टू इंक्रीज इट्स प्रोडक्टिविटी एंड यील्ड, एंड इट इज नोन एज CO2 फर्टिलाइजेशन इफेक्ट. ये वाला इफेक्ट हम C4 प्लांट्स में नहीं ऑब्जर्व कर सकते ऑब्वियसली, क्योंकि उनका रिक्वायरमेंट फुलफिल हो रहा है. राइट, एंड इफ वी टॉक अबाउट द टेम्परेचर, so in C3 plants the optimum temperature for them is 25 to 30 degree centigrade. ये इतने ही टेम्परेचर पे सर्वाइव कर पाते हैं. फिर उसके बाद दे विल स्टार्ट फोटोरेस्पायरिंग. फोटोरेस्पिरेशन होना उनमें स्टार्ट हो जाएगा. अगेन दैट मीन्स उनकी प्रोडक्टिविटी और यील्ड कम हो गया. अगर हम C4 प्लांट्स की बात करें तो वहाँ पर इनका जो टेम्परेचर ऑप्टिमम है इट इज़ अराउंड 35 to 45 डिग्री सेंटीग्रेड. यानी कि ये ज़्यादा टेम्परेचर में भी सर्वाइव कर सकता है बहुत आराम से बाय कॉन्सेंट्रेटिंग द CO2 इन द बंडल शीट सेल, एंड दैट्स वाई द प्रोडक्टिविटी एंड यील्ड ऑफ द C4 प्लांट इज मच ग्रेटर देन द C3 plants. So these are the concise difference between the C3 and the C4 plants. Okay, now let's make a table and jot down all the key points which we have studied in the class and make the complete differentiating table. Let's make. यहाँ पे हम लिखेंगे C3 plant. हमने कुछ डिफरेंस पहले भी किए थे. अब जो फैक्टर्स अफेक्ट करके जो डिफरेंस हुए इन प्लांट्स में वो हम लिखेंगे. So if we talk about the temperature optimum in C3 plants, it is 25 degree to 30 degree centigrade, and if we talk about the C4 plants, so it is around 35 degree to 45 degree सेंटीग्रेड. राइट, अगर हम CO2 की बात करें, CO2 कॉन्सेंट्रेशन की, सो इट्स सैचुरेशन इज बियॉन्ड 450 माइक्रोलीटर पर लीटर. एंड इफ वी टॉक अबाउट द C4, इट इज 360 माइक्रोलीटर per liter. That's why CO2 is the major limiting factors in the rate of photosynthesis. हमने light के बारे में पढ़ा था, which is rarely a limiting factor. So if we talk about the CO2, it is the major limiting factor for affecting rate of photosynthesis. Temperature पढ़ लिया, carbon dioxide concentration भी देख लिया. Right, and if we talk about the photorespiration, so in C3 plants, the rate of photorespiration is high. But if we talk about the C4 plants, it do not take place in them. Right? Again, the number of ATP which we have learned and the number of NADPL which is similar in the C3 and the C4 pathway. So these are the complete differences between the C3 and C4 pathway. So here I can see that C3 is cool, confused and common, but C4 is clever and champion. अगर हम C4 plants की बात करें, for example, our maize plant, which is actually clever and champion. Why so? Because it have done the bifurcation of work and it has fixed the carbon dioxide in the another cell rather than in the mesophyll cell. But in C3 plant, it has only one cell to assimilate the carbon dioxide. That's why it is cool, confused, and common. Now, let's see the previous year questions which are asked from this topic. Let's see. The first one is from the year 2023. As I've told you, इस topic से almost हर साल questions पूछे जाते हैं. Now, let's see. Yeah. Let's see. Yeah, the first question is given below are two statements. So it is statement based question. The first statement states that Rubisco is the most abundant enzyme in the world. So yes, this is absolutely correct statement. Rubisco is the most abundant enzyme on this planet. And if we talk about the most abundant protein, please tell me that what is the most abundant protein in the biosphere. Please write down in the comment section. Now, let's see the next statement which says photorespiration does not occur in C4 plants. So yes, this is absolutely correct statement. We have studied that C4 plants में photorespiration नहीं होता. So here both statement 1 and statement 2 are correct and it is correctly depicted in option 1. Right? So this is how we rule out the other options as well. Now next question which says when one CO2 molecule is fixed as one molecule of triose phosphate, which of the following photochemical made high energy chemical intermediates are used in the reduction step? जैसा कि हमने C3 pathway और Calvin cycle में देखा था that it consists of 3 steps. First one is carboxylation, next one is reduction and third one is regeneration. Right, तो reduction step में हमारे कितने assimilatory part use हुए थे for the single CO2 molecule? Those are 2 ATP along with 2 molecules of NADPH and it is correctly depicted in option 1. Rest option gets ruled out. Again, this is question from the NEET 2022. Now, let's move ahead with the next question which says, which of the following statements is incorrect? इसमें से कौन सी statement गलत है? Let's see. The first one is Rubisco action requires ATP and NADPH. So yes, it is absolutely correct. Rubisco enzyme, which is the most abundant enzyme on this earth, most abundant enzyme in the world, require assimilatory part to work. Those are ATP and NADPH. तो ये statement हमारी correct हुई. Now let's see the next one which says Rubisco is a bifunctional enzyme. Yes, it is also correct. Why? Because if we expand the name of the enzyme itself, that is Rubisco, ribulose, bisphosphate, carboxylase and oxygenase, then we can see that yes, it is the bifunctional. इसके पास दो activity हैं, oxygenase की भी और carboxylase की भी. तो ये statement भी correct हुई. Next one is in C4 plants, the site of Rubisco activity is mesophyll cell. तो हमने जब differences देखे थे, we have seen that action of Rubisco enzyme in the C4 plant is bundle sheath cell, where the assimilation of carbon dioxide is taking place and the activity of Rubisco enzyme is present in them. But if we talk about the mesophyll cells found in the C4 plants, in them PEP carboxylase enzyme is present, but Rubisco enzyme is absent, right? So this statement is incorrect. क्योंकि mesophyll cell में Rubisco enzyme होता ही नहीं है. Next one is the substrate molecule for Rubisco activity is a 5-carbon compound. So yes, it is absolutely correct. If we talk about the C3 pathway, the substrate compound is RUBP, which is 5-carbon compound. In this question, we have to see the incorrect statement. Therefore, the correct answer is option 3. Let's move ahead with the next question that is from the NEET Phase 1 2022. Now let's see that what is the role of large bundle sheath cells found around the vascular bundles in C4 plants. C4 plants में एक बहुत ही beautiful anatomy होती है which is known as Cran's anatomy, which is also known as wreath arrangement, where the vascular bundles, I am writing VB for that, are surrounded by bundle sheath cells like this. They are tightly packed. जैसा कि आप देख सकते हैं कि इसमें ring like arrangement होता है. They have thick walls, abundant amount of chloroplast, they are impervious to gases. That's why CO2 इसमें से बाहर नहीं जा पाता और concentrate होता रहता है for the Calvin cycle. And later on it is surrounded by mesophyll cells. So this is the Cran's anatomy which is found in the C4 plants. Now let's see the features. To enable the plant to tolerate high temperature. हाई टेम्परेचर को टॉलरेट करने के लिए Cran's anatomy का कोई रोल नहीं है. टू टॉलरेट द हाई अमाउंट और द हाई टेम्परेचर द रोल ऑफ स्टोमेटा इज देयर. ओपनिंग उसकी क्लोजिंग जल्दी हो जाती है. इट डू नॉट अलाउ स्टोमेटा टू ओपन मोर टाइम. सो फर्स्ट इज इनकरेक्ट. Next one is to protect the vascular tissue from high light intensity. Vascular tissue already embedded है in between the mesophyll cells, तो वहाँ पे light intensity से protect करने के लिए bundle sheath cell का नहीं requirement है. Third one is to provide the site for photorespiratory pathway. Again, bundle sheath cell में कोई भी photorespiration जैसी चीज़ नहीं होती है, because photorespiration require three different organelles which are chloroplast, mitochondria and peroxisome, but bundle sheath is not involved. So third option भी incorrect हो गया. Now let's see the fourth one, which says to increase the number of chloroplasts for the operation of Calvin cycle. So yes, it is absolutely correct. To increase the concentration of carbon dioxide in the C4 plants, bundle sheath cells are present by increasing the number of chloroplast in them, ताकि ज़्यादा से ज़्यादा Calvin cycle हो और ज़्यादा CO2 fix हो पाए. Right? Therefore, for the question, the correct answer is option 4. Now, let's see the last question, that is the oxygenation activity of Rubisco enzyme and photorespiration lead to the formation of. यहाँ पे photorespiration का concept आ गया. This question is again from 2020. So, when photorespiration takes place, that means Rubisco enzyme is acting as an oxygenase. That means in the presence of oxygen, it is acting as an oxygenase and it will lead to the formation of two compounds. One is 3-carbon, that is phosphoglyceric acid, and another is 2-carbon molecule, that is phosphoglycerate. Right? Now, let's see the option. The first one is one molecule of 3-carbon compound. So, yes, it is absolutely correct. One molecule of PGA is formed during the photorespiration. Let's see the other options as well. Next one is one molecule of 6-carbon compound. तो 6 कार्बन का तो कंपाउंड बन ही नहीं सकता because Rubisco खुद 5 कार्बन का है और जब वो ऑक्सीजन के साथ कंबाइन हुआ तो उसको कोई एक्स्ट्रा कार्बन नहीं मिला. Okay, so second option gets ruled out. Third one is one molecule of 4-carbon compound and one molecule of 2-carbon compound. Again, अगर हम इनको add करें तो 6-carbon compound बन रहा है. तो again, Rubisco के पास 5 था और ऑक्सीजन के पास कोई कार्बन नहीं है. So this is also not possible. Fourth one is 2 molecules of 3-carbon compound. Again, this is incorrect. For the question, the correct answer is option 1. So students, so this is all about the C3 and the C4 plants. I hope this lecture will help you to strengthen your concept on these two plants, and this will help you to understand that how we can improve our crop and how the changing environment is affecting our crop plants. So thank you for watching this lecture. Thank you.